If you look closely at raw RNA sequencing data, it is a nearly unreadable wall of text. But to make informed decisions about your research, you need that chaotic text to look like this: a clean, interactive dashboard. By the end of this module, you are going to generate a comprehensive quality control report that provides a clear, quantitative baseline for the health of your sequencing data. To follow along, you need three things: active NIH BioWolf credentials, a secure NIH network connection, and basic Unix command line navigation. Verifying your data's health before you begin complex analysis is crucial. Taking these steps prevents you from wasting compute time on compromised samples. The resources required to process genomic data are provided through high-performance computing systems here at the National Institutes of Health. To tap into that infrastructure, open your terminal and establish an SSH connection to BioWolf. Once you are in, stop. Do not run any analysis right here on the login node. This is a shared entryway for hundreds of researchers, and running intensive processes here can slow the system for everyone. Instead, we move to a private workspace. Type the command sinteractive and hit enter. This command secures a dedicated compute node, giving you the resources needed to run your tools. Now that we have room to work, make a new dedicated directory for this specific module. Next, copy the provided class data, specifically the HCC 1395 FASTQ files, directly into this newly created folder. Type ls to list the directory contents and confirm your files successfully transferred. Isolating your data and compute resources helps ensure your environment remains organized and your analysis results are easier to track as your project grows. We are going to analyze these sequence files using FastQC, a standard tool used to calculate quality metrics for raw sequencing data. Because bioinformatics tools are not active by default on BioWolf, you must explicitly load it into your environment. Type module load FastQC. With the module loaded, type fastqc star.fastqc. The asterisk acts as a wildcard, commanding the software to process every single sequence file in your directory at once. Press enter. You will see lines of text begin to populate the terminal, showing progress percentages as FastQC methodically processes each individual file. Once it finishes, list your directory contents again. You will see that FastQC has generated a new HTML and zip file for every sequence. We have the diagnostic data, but opening and reviewing dozens of individual HTML files makes it difficult to see how different samples compare to one another. To fix that, we use an aggregation tool called MultiQC to merge those individual outputs into a single report. Load the MultiQC module into your current session, just as we did before. Now type multiqc followed by a space and a single period. That period tells the tool to look for data within your current directory. Hit enter. MultiQC finds the FastQC logs you just created and compiles them into a unified report. Looking at this output, we can see how it pulls in metrics like mean quality scores, GC distribution, and sequence lengths for the entire dataset. The easiest way to view this graphical HTML file is to open your local web browser and navigate to the HPC on-demand web portal. Using automated tools to aggregate these reports provides a holistic view of the dataset, making it much easier to spot trends or anomalies across dozens of samples. With the report open in your browser, start at the top. The sequence counts and summary statistics show you the total number of reads and the overall file sizes for each sample. Next, scroll down to the per-base sequence quality graph. This chart categorizes the data into scoring zones. Reads falling into the lower red zone represent poor quality, while those in the upper green zone indicate highly accurate reads. Moving further down, check the adapter contamination heat map. Any bright red spots here indicate lingering artificial sequences left over from the laboratory process. You now have objective proof of your dataset's baseline health. These contamination metrics tell you exactly what you need to target as we begin cleaning and trimming the reads in the next lesson.